life process. And so this slide, just a schematic, shows for colon cancer or colorectal cancer, there are at least about six morphologic stages, pathologic stages that we can identify, going from a small, well, initially there are things you can only see microscopically in tissue. Then you have small polyps that progress, develop dysplasia, and become cancer. Now, it's well known that there are molecular events that underlie this. And so for colon cancer, there are two main pathways. There are actually more, but there's one called this chromosome instability pathway. And then there's microsatellite instability, or SIMP. Don't worry about the details, but the main point is that they have specific mutations that progress in a, they're not always exactly in this order, but there's a preferred order. So for example, the APC mutation is almost always an early event. So that's one of the first things that happens for colon cancer. So the cancers progress through these stages. That's just the main point. Now, this slide is just a summary. This was actually a paper that just came out, which was kind of timely. Um, from around 1950, there were studies that suggested this initiation, promotion, uh, progression for cancer. And the concept is, and this was done in animal studies in the 1950s, where you give a carcinogen, so something that causes DNA damage, and which is called initiation. And then, but so you can have many, many cells that have a mutation, uh, but they don't progress to cancer typically unless a promoter is given. And a promoter is typically something like, I mean, here's just a schematic picture. That could be like pollution or smoking, obesity, probably hormones related to obesity, like estrogens for breast cancers could be promotion. And then this is picture's a little obscure, but it's usually inflammation. Anything that causes chronic inflammation in the tissue will be a promoting factor. So basically, what you have is you start with these initiated cells that can happen, perhaps early in life, but they don't progress to cancer unless you have this long-term promotion, um, and which could be related to lifestyle and doesn't have to be a carcinogen. In fact, it usually isn't directly DNA damaging. It could be things like inflammation or hormones, things like that. And this is a photo from actually a commentary from this. A study that went back, was published just a few weeks ago, that looked at the initiation promotion model, and what they really added is that they can actually, the initiation promotion model is very empirical. Like they really didn't look at DNA specifically. In this model, they for example, they could show once they gave the promotion, I'm sorry, the initiating carcinogen, which is DMBA, to the mice, they could see that they had thousands of cells that had mutations in, for example, RAS oncogene, which is a driver mutation. So all you have thousands of cells that have driver mutations, but interestingly, they don't get cancer unless they have this promoter effect. Like obesity was one of the things that promoted uh, progression of the cancer. Now, so that's animal data. Now, in human data, you can't obviously do studies like that. But in some ways, unfortunately, obviously, we do have some, natural or man-made experiments, I guess you can call, that show probably similar processes that are going on. So there have been many studies of radiation, like clinical radiation, and then there's also studies of follow-up from the atomic bomb survivors from Japan. Uh, so, and in this case, it's interesting that you know exactly when the initiating event happened. It happened in a millisecond. So the population got exposed and had DNA damage, and then people were followed for a long time. And you could see that. So this slide's a little complicated, but the y-axis shows excess cases per 100,000 person years at this people that got this estimated exposure uh, to radiation. And so if you look at people that were at age 10, this is for women and for men. So they were age 10 when they got the exposure, one-time single exposure. And you can see even at age 70, there's an excess of rates. Um, and what's kind of interesting though is that um, if you look, so for example, let's just focus on the women. Uh, this are the rates of excess cancers for those exposed at age 10. So if you look at age 50, this is their rate. And if you look at uh, this one is for women who were exposed at age 30, and their rate at 70 is just about the excess number of cancers is similar to the ones that were exposed at age 10 and age 50. So if you go 20 years later, so they were exposed at 20-year later age. So they basically get the same history of cancer, but of course everything starts 20 years late. So of course the people that were exposed earlier just have many more years to accumulate more cancers. But if you go from the starting point when they were exposed, they actually have the cancers they get are pretty similar in number, the excess cancers. Um, this was another tragic thing that happened. This was in Chile in 19 the water supply in this region uh, was exposed, of course, unknown to high levels of arsenic, which is a carcinogen. So the exposure happened from 1959, and then in 1970, they figured out what was going on, and that's when the exposure stopped. So these are rates of bladder cancer in the population. So this is a time period of exposure. So over this time period, you can think that there was damage happening to cells in the body. In this case, looking at bladder cancer, and you can see that cancer rates didn't go up right away. Exposure starts at 59, and then about you know almost 20 years later, rates start going up, and interestingly, you can see that rates maintain above almost twofold higher than the other the unexposed population, even like up to 2010. And so that you have this one-time exposure, and then you have this maintenance of high rates of cancer many years later. So so that's a little bit of background. And the next point I want to make is that it takes many years or decades for the cancers to progress. And I mean, an obvious, I'll just give you a few examples of this, and I just showed this. So this is a good example where you can see in the arsenic exposure that, like, for example, there are some people that were, let's say, initiated at age 15. And 45 years later, they're still at higher risk of cancer at age 60. So this example shows that you can have many, many years after the initial exposure where the cancer is elevated. There are many examples. This is one of the first studies I did many, many decades ago, showing my age here, that it had been controversial whether smoking was related to colorectal cancer. And initial studies, like the classic smoking studies that were done, like in the 1950s and 1960s did not show a link with smoking and colorectal cancer. Did obviously show smoking associated with other cancers, particularly lung cancer. But um, we hypothesized that it just would take longer to see an effect on cancer. Now, smoking started in the early 1900s. In this study, in the nurses and health professionals cohorts, these are studies in Boston, we could see that you don't see a rise in cancer until 35 to 39 years after the individual started smoking, and then the rates go up. And what's interesting is that rates remained elevated even if they stopped smoking. Um, Now, this slide, sorry if it's a little confusing, but uh, I thought this was interesting. That this is more of a modeling study, and so what they did is, uh, well, I won't go into detail. It's actually a pretty complicated model, but this is based on uh, mutations, and they estimated how many mutations in different tissues, how long they take to progress to cancer. And these are estimates. They assume the three driver mutation model for these cancers. Um, so I'll focus on colon, but you can see the it's pretty similar for other cancers. Leukemia actually only needs two driver mutations in this model. So what this shows is um, so this T1, T2, T3. Um, it shows like assume there are three driver mutations. So cancers have to acquire these three mutations, and they estimated how long it takes to go through each one of these stages. So T1 shows about 15 years. And this is from uh, birth to to age 15. And then of course there's a range, but that's the average. And then after that, so these are things that eventually become cancer. The second stage would take about almost 20 years, and the third stage about 30 years. So if you add these up, they add up to like about 65, which is the average age of colon cancer, roughly, of getting cancer of the colon. But what this shows pretty dramatically is that the first mutation on that for people who eventually get colon cancer, the first mutation, the average age occurred at about age 15. So we're, in childhood, adolescence. But then it takes many more years for the cancer to fully develop. And this is actually very consistent with just the empirical incidence rates that we see in cancer, like for colon cancer. So just keep that in mind that at least based on modeling, the first mutation for colon cancer occurs around age 15. Um, okay, so now I haven't really talked about susceptibility uh, of getting cancer. I mean susceptibility for the exposure to induce, like let's say, a mutation at a certain age. And so I've just talked that regardless when the initiation starts, it takes a long time to get cancer. But another question, a related cancer is there are you just more susceptible at early age to an exposure? Uh, and there's some, examples where it's definitely true that, and one classic example I'll just mention briefly is hepatitis V um, exposure, which is a big, determinant of liver cancer. And the age of hepatitis B virus exposure largely determines the incidence of becoming a chronic HBV infection, which is what's required to eventually lead to liver cancer. So, but if you get the infection in adulthood, the vast majority are cleared, so it's not a problem. But it's really a problem at very early ages, like neonates, very high rates, and children, 30% age 1 to 5 fail to resolve HBV and develop chronic infection. So this is a case where the carcinogenic exposure is HBV, but the age that you get it is, the main determinant whether you'll get cancer. And it's probably related to things like the liver development or the immune development of the immune system. So it's just that younger age is more susceptible. Um, now, that's for liver cancer, but I think there's some exposures that are important for cancers broadly. And a couple of them are very like basic physiologic processes. So the rate of body growth, example, height, which can be a surrogate, is associated with a majority of cancers, and also events like menarche, which I'll talk about. But this is a slide, a big population study in Korea, but this is shown in many other populations, very consistent finding that being tall is a risk factor for cancer. Um, and so if you look at total cancer, for every if you look at population, 5 centimeter increase in height, and this is in non-smokers, it's associated with a 10% increased risk of all cancers combined. And as you can see, almost every cancer is increased, some more than others, but almost every cancer is increased. Pretty remarkable finding. What may account for this? Well, we don't know for sure, but th this slide shows the age pattern for insulin-like growth factor 1, IGF-1, which is a potent growth factor. In fact, it's one of the determinants of height. So if you're very high in this, you can be very tall. And if you're short, you can have, you'll be on the shorter end. But what's interesting, I mean, we have IGF-1 levels that you can measure throughout life. And if you get a measure, actually, even one measure in life, it does predict your risk of certain cancers like prostate, breast, and colorectal. So even a single measure at one point in life increases risk. But what's interesting is you can see that during the growth period in adolescence, you get 2 to 3 times higher level. So this is a period where there's a lot of cell growth going on, and we think that that's probably why it's a period of high susceptibility for cancer events. Of course, cancers don't occur at that age. It's just that the initial events are probably occurring at that age. Another well-known factor, and probably most people know this, age of menarche. The earlier this is from a large pooled analysis. The earlier the age at menarche, the higher the risk of breast cancer, well established in many studies. So these very early events are important for cancer much later in life. Now, what's interesting is that for breast cancer, early age of menarche is a clear risk factor shown many times over. In prostate cancer, we always suspected that early age of puberty would be a risk factor for prostate cancer for similar reasons. But it's very, you don't have a clear marker for onset of puberty on a population level in boys as easy as age at menarche. But this was a Mendelian randomization study done about 10 years ago that showed pretty clearly that the later the onset of puberty, based on the Mendelian randomization, so the genetic predisposition, the lower the rate of prostate cancer. This slide is kind of it's an interesting format. They show cancer risk reduction. So this This is later onset of puberty, and so you have like about a 20% lower risk of prostate cancer. So it's probably the same processes that like the things that are going on at puberty, as we know, are very important for breast cancer, and it's probably the same in prostate cancer. Um, this just summary. I wrote a review with Ginny Her a few years ago, summarizing the data for this, and it shows that earlier onset of puberty and longer time in puberty is associated with increased risk of prostate cancer. The mechanisms are not definitive, but it's probably what we think, at least, it's high exposure to testosterone and IGF-1 in prostate tissue. The earlier age at puberty, ha keeps you in a longer time of exposure to these growth factors. And it's interesting that there's data to support that this pattern can largely explain some of the differences that we see. So, like African Americans have high rates of prostate cancer, and they tend to have early age at puberty. And the Asians have later age of puberty and have lower rates. So it doesn't prove cause and effect by any means, but at least it's consistent. And it's also interesting that we know that the differences that we see in African Americans and Asians, uh, those differences in rates do start early in life because you can see the early lesions of prostate cancer, even in the man's like in their 20s, you see the pattern for African Americans, whites, and Asians. So it's something that, of course, the cancers are diagnosed at older ages, but the process almost definitely starts at an early age. So before the last part of my talk, which I'll show some of our data, uh, this is just a summary slide, and this tries to summarize. I hope doesn't cause more confusion of the main points. Really, two main points that I've been trying to make. So schematically, what we have here is this is just the life course from birth to 100. And then this just schematically shows exposure, whatever diet, obesity, carcinogens can occur, at any point in life. Um, and uh, but but cancer risk, which I try to show here, tends to occur later in life. Of course, there's some unfortunate cases at early ages, but most cancers are aged after age 50, probably over 90%. So these are the adult onset cancers. So now, what could be happening? Um, like, why could early life exposure be important? And I think there are two main reasons. The first is, as I just summarized, that maybe there's some heightened organ susceptibility at early ages, like, illustrated, for example, like age at menarche for breast cancer. So that's something that's occurring, during puberty, adolescence. And so that period inherently makes you at higher risk to at least start the cancer process. And then I also showed some example like with HBV. There are other potential examples, like the microbiome is probably the first few years of life is where it's important, where you're exposed to certain bacteria, and that starts the gut microbiome, for example, uh, to get that will maintain a pattern throughout life. So there's susceptible times at early. Uh, but then another big reason why early age is important is a very practical, simple reason. It's not theoretical. It's just that um, if the process starts earlier, you just have more time to get the cancer. And what I show here very schematically is, let's say the asterisk is when you have, for example, the first mutation. That's where initiation starts. Uh, and um, and then it takes 50 years. Now, of course, there's going to be a range, but let's say on average in this simple model, it takes 50 years to develop cancer. So this person happened to get a mutation, around age 10 or 15, and then by 60, they develop cancer. So these persons got the same mutation, same process, and they got cancer later. And then the last one is not shown, but I have a couple of cases here where a person got the same initiating mutation and they were in the process to cancer, but they died at age 90. Uh, so, so that's why, in a sense, you can be getting mutations at any point in life, but it's just that if you get it earlier, you're more likely to get a cancer within your lifetime, and obviously more likely to get a cancer at an early age. If someone has cancer at age 50, it's likely the process started at much earlier age. If someone gets a mutation at age 50, then even though they're still at risk for developing cancer, they may not have enough years left in their life. So competing risks will prevent them from getting cancer in a sense. So it's just a timing thing. So these are two main reasons why early age may be important. So finally, I'm going to show some examples, at least focusing on lifestyle factors. There are things like let's say radiation, like clinical use of radiation, for example, that clearly show a risk factor for cancer. So there's no doubt that early age could be an initiating point for cancer, but there's less evidence for modifiable factors like diet. So I'm going to show some emerging examples for colorectal and breast cancer. And most of the data on this is actually from one cohort. And it's the Nurses' Health Study 2, which is an ongoing prospective cohort of women. And the cohort started in 1989. And an interesting thing about the cohort is that uh, they were relatively young, age 25 to 42. Many cohorts like nurses, one, health professionals, there are many cohorts now, they typically start much older in life. And in 1998, which is about 9 years after the cohort started, there was a questionnaire sent asking about diet in their high school years. And uh, there's validation studies that I won't get into here, but we can talk about it later if we want. But of course, the data are far from perfect. But there is, it's not as bad as people might think. There is some predictive ability of the women recalling their diet from adolescence. Like the average correlation in this study was about 0.58, where the women recall their diets later in adulthood, their high school diet. Um, uh, and they had by chance that they had in this study they had records of their diet at age high school. So they already had data for this. But unfortunately, there are not many studies where you actually have data. But at least this study was able to show that there's some ability for people to recall their diet. Um, Now, what's also interesting, that there have been other studies that look that the older and older you get, the worse the recall is. So the Nurses' Health Study is really, to my knowledge, the only study, Nurses Study 2, sorry, is the only study where you have relatively young women with a comprehensive questionnaire recalling their diet. So we could start looking prospectively at cancer. And just one kind of general point I wanted to make, so I actually made a slide to make sure I make this point, is that like early life exposure is often correlated with adult exposure. There could be many reasons, bad habits or good habits can start early in life and progress to adulthood, or things like, obesity. There are genes for obesity. So individuals that are, tend to have obesity early will also tend to have it late. So there's this correlation. And, to one extent, if this is a causal association, so, you can say, starting a habit early in life causes an adult exposure. And let's say the adult exposure is associated with cancer directly. So that's where the etiologic time, you can still make a case that this is relevant for carcinogenesis, even if early life itself is not the causal factor. It's just that it, causes, in a sense, a predisposition to be exposed in adulthood. But it's also possible that early life exposure is the main risk factor. And that's mostly what, I mean, the first part is important, obviously, from a public health perspective. In other words, you might want to start good habits as early as possible. But from an etiologic perspective, I'm more interested is, does early life exposure directly cause cancer risk? So I'm just going to present quickly some results that we have for colorectal cancer. And for colorectal cancer, it's the cancer that actually has the most modifiable risk factors. It's very closely related to life. And these are adult factors: tobacco, alcohol, obesity, physical inactivity, red meat, increased risk, and prudent diet, fiber, and dairy and calcium are associated with decreased risk. These are well established in many, many studies. So, our group started to look at these factors for beginning at early life. And so I'll just go through some of our findings relatively quickly here, but I think you'll get the point. So this is BMI at age 18, and then weight change since age 18. And you can see that, uh, and these are modeled simultaneously. You see that BMI at age 18 is associated with an increased risk of colorectal cancer, and also weight gain after 18. So both, have your early weight independently and your weight gain later in life, both increased risk or associated with increased risk. Another factor is physical activity. And some of these studies are for adenomas, which are well-established precursors for colon cancer. So they're a surrogate, particularly advanced adenomas. So these are the large dysplastic adenomas. So and actually that's probably a better endpoint for colorectal cancer. But so in this slide, we look at physical activity. The reference group is low physical activity in adolescence and low in adulthood. And these are people that are high in adolescence, high in adulthood, and they have almost a 40% decreased risk of adenoma. But what's interesting is that if you're high physical activity in adolescence and low in adulthood, you also have a 21% decreased risk. Not as strong, it's better to maintain physical activity throughout your life, but this slide shows that you do get some impact from being physically active, even just in adolescence. Another thing is smoking. And in this slide, we look at smoking before the age of 30. So these are uh, uh, these relative risks are for a 20-pack year increment. And this is simultaneously adjusted for smoking after age 30. And you can see that for adenomas, large adenomas, and cancer directly, the smoking early in life is actually more predictive or stronger predictor, even though smoking after age 30 also is associated with an increased risk, not as dramatically. In fact, for cancer, it's not increased. So it's almost like you start the smoking you do before age 30 is more important for colorectal cancer than after age 30. Uh, Diet quality, and again, this is from the questionnaires, and we do see Western diet pattern associated with large colorectal adenomas. And for villus adenomas, that's a type of adenoma that's more likely to progress to cancer. Uh, um, this is looking at sugar-sweetened beverages, which increased risk. What this slide shows actually is it's called a substitution analysis. So these are substituting these drinks, artificial sweetened beverages, coffee, milk, or total milk. Uh, Drinking these are associated with a reduced risk of early onset colorectal cancer. So sugar sweetened beverage intake early in adulthood and adolescence is associated with increased risk, and things like milk is associated with a decreased risk. This is alcohol, and in alcohol we don't we well they may be having it in high school, but this is asked in age 18 to 22. We show that alcohol high in early adulthood and low in middle adulthood is associated with an increased risk. So even if you stop drinking, you already incurred some increased risk from drinking early in life. So this is quickly summarizing what I just showed. In Nurses Health Study 2, early life lifestyle. So this essentially childhood, adolescence, and age 18 to 21 is associated with colorectal adenoma or cancer risk, very similarly to adult lifestyle. Uh, Adverse is tobacco, alcohol, obesity, Western diet, excess sugar. Beneficials, physical activity, prudent diet, and dairy, calcium. So you see the same pattern early in life, independently as late life that associated with increased risk. So quickly for breast cancer is a little bit different from colon cancer in that for colon cancer, you might recall that there were many risk factors in adult life that increased risk. For breast cancer has been studied so much for diet, and there's very little evidence for diet in middle age and older women being important. Obesity, postmenopausal, is definitely increases risk, but diet per se, independent of obesity, has not been shown to be an important risk. Now, what's interesting for breast cancer, and I just have a slide here. I think I showed earlier the atomic bomb cohort, and for breast cancer, more so than other cancers, it seemed that early life exposure is much more important, even accounting for the follow-up time. So for girls that were exposed before age 20, the excess risk is very high. And then exposure after age 40 or so, even with longer follow-up, does not seem to be much of a risk factor for breast cancer. For breast cancer, early life exposure, tissue sensitivity does seem to be important. And there are only a few studies that have been done for breast cancer based on in the Nurses Health Study 2 with the adolescent diet. Um, um, and this study shows that substitution of poultry, legumes, nuts, and fish for red meat in adolescents is associated with lower risk of premenopausal breast cancer. So you see the protective association. This is all women, and this is for premenopausal, not so much for postmenopausal. Another thing is fruits. This is fruit of various fruits, and we see that a number of them, including, for example, grapes, bananas, apples, uh, some are suggestive that are associated with reduced risk of breast cancer. But early adulthood fruit intake is very null. So fruit seems to be protective, but only the fruit consumed during adolescence. Even early in adulthood, once you get beyond age 20 or so, doesn't seem to show much. Um, and this shows fiber. Um, and again, in adolescence. Um, and you can see that fiber is has an inverse association, particularly for premenopausal, less apparent for postmenopausal women. And finally, these are adolescent dietary patterns. This is a prudent dietary pattern, and it shows for all women, probably driven mostly by premenopausal women, an inverse association. So in Nurses' Health Study 2, adolescent diet appears associated with breast cancer, particularly for premenopausal breast cancer. Um, and so specifically a prudent dietary pattern and diet high in fruits, fiber, and low in red meat are associated with a decreased risk. Interestingly, in adulthood, these factors do not appear to be associated with breast cancer. So this is my last slide. And so basically, what I tried to show is that multiple lines of evidence demonstrate that many cancers are initiated in childhood and adolescence. And there are two main reasons. One is some tissues have a heightened sensitivity to cancer at certain time periods, like puberty. And the other reason, a very practical reason, is that if you start early, you just have more time for a cancer to develop. Uh, and of course, these are very early data that need to be replicated, but it's interesting that colorectal cancer seems to follow pattern too, in that diet and lifestyle seems to be important throughout the life course. So even adult risk factors are important, but early risk factors are also important and probably will be even more important for earlier onset cancers. You just start the process early. So there's no special biology at early age being important. It's just that you start to, things earlier. Breast cancer seems to be more related to like a pattern one, which is you specifically have an important sensitivity, and then later in life, you lose that sensitivity. So for breast cancer, maybe particularly early in life and adolescence, before age 20, where exposures are really important for breast cancer. And again, that's not to say that there aren't some exposures like promoting factors late in life, like obesity and postmenopausal breast cancer that aren't important. But a large component of breast cancer will also be related to early life exposure. So I wanted to leave some time for questions. So I think we do have, I think, 12 minutes or so, or so I'd be happy to take any questions at this point.
Thank you so much for this talk, Ed. And let's open it up. Let's open it up for questions. You can either, of course, come to the front and ask the question yourself, or I'll quickly repeat it on the microphone. Whom can I give the floor? And of course, we're watching online as well if you have questions there on.
So maybe I start. Thank you very much for this wonderful presentation and an overview. And I think for the audience here, the topic was extremely interesting because we are leading one of the child cohorts in Europe, multi-center cohort in children, adolescents, and we study many of these factors, and we see importance of lifestyle naturally with regards to metabolic dysfunction, early onset cardiometabolic diseases, and now it's really interesting to trace that to such devastating outcomes such as cancer. But the question here is, how do we outline the difference between early onset and late onset cancers? And we know that, for example, you mentioned breast cancer as a composite outcome, but we also see, and we have seen in your slides, a number of differences between pre and postmenopausal breast cancer. We know, for example, that obesity is detrimental and is an important risk factor for postmenopausal breast cancer, but not for the premenopausal breast cancer. So what would be some suggestion for us to, if we want to conduct research in the future, how we should outline these differences according to outcomes?
Yeah, thank you. It's a great question. Breast cancer is always complicated. And so obesity early in life is associated with a decreased risk of both pre and postmenopausal breast cancer. And the reason is not clear, but it's very definite. Even there are Mendelian randomization studies that support this. So it's probably somehow related to something going on with hormones, like obesity might be associated with lower IGF. It may impact the estrogen exposure early in life. Uh, so obesity, and obviously there's not a public health message here. I wouldn't recommend obesity as a protective factor for breast cancer, but it's important to know. Uh, so now later in life, postmenopausal women, adipose is the main source of estrogen. So that's what drives uh, postmenopausal breast cancer. They're mostly estrogen receptor positive, and the estrogen is a big driver. So there's a very, practical message for preventing postmenopausal breast cancer. I would definitely focus later in life, postmenopausal. For premenopausal breast cancer, at least the emerging data that we have